Good afternoon, everyone. It is my great honor to be able to introduce our next speaker. She is unabashedly Canadian. I believe she may be the only Canadian keynote speaker here at this conference. Am I right? Well, if you have to pick one, you've made a good choice. And if you want to know if the Toronto Raptors are on a winning streak, be sure to follow her on Twitter. Within the realm of science, though, she's known for performing some of the most interesting and exciting scientific studies on the cellular basis of learning and memory, particularly with respect to the amygdala. Her work is one of the reasons why I don't use a textbook for my undergraduate teaching. Why? Well, if you go to the textbook section on fear learning, you read about classic work showing the amygdala involvement in aversive learning and so on, which is great. But students at this point have also learned the basics of neurophysiology and its theoretical implications for learning and memory. But it's still largely theoretical in the textbook. They're left wondering, well, is there any solid demonstration that LTP is directly involved in memory, or that cellular excitability is involved in encoding processes? And these are the questions that we all feel like there should be an answer for. And actually, thanks to her, we're actually getting these answers. It's just that the textbooks have not caught up. I'm talking about things like showing that neurons are not randomly selected for memory encoding, but they're selected based on their excitability state, that you can artificially make memories appear and disappear. And so this is who I go to when I want to teach students about the latest and most inspiring research in the field of memory, the kind of stuff that just a few years ago could only be found in the movies. Now, I'm a scientist, and so I'm never happy with the sample size of one. And what I've told you so far are just my observations about her and her work. And so to get a better sample size, I reached out to folks who have passed through her lab to see what kind of a mark she's left on her trainees. I haven't counted the number, but there are many that have passed through her lab and gone on to establish their own successful research careers. And while everyone had a universal appreciation for her sense of humor and her scientific prowess, one thing that stood out to me was the impact she left on her female trainees. They all spoke to the instrumental role she's played in mentoring them in a field that's been historically dominated by male scientists. When her lab was still relatively young, she had her first child. And whereas that can be a turning point where many junior scientists, particularly women, may make a choice between caring for their child and caring for the quality of their research and the quality of their training program, the productivity did not dip one bit. But to do this, she had to do things differently. Raising a child became part of the atmosphere of the lab, and it probably made a lot of people uncomfortable. But had she not done it, she might not be here today to deliver the amazing talk you're about to hear. Had she not done it, we might not be benefiting from the next generation of female scientists that have passed through her lab. She's courageous in her science, but she's also courageous in changing the culture of science for the better. And so with all that, let's thank and welcome Sheena Josselyn.
Wow, thank you so much, Jason. That was a lovely introduction. And I want to give a shout out to Mike and Manuela for putting together this amazing meeting. And happy 40th birthday to UCI Learning and Memory Center. You don't look a day over 39. So I'm going to be, as Jason mentioned, talking to you about my work on how memories are formed and stored in the brain. That's really been the sort of question I've been asking since I got into this biz. So the idea that memory is stored as an enduring change somewhere in the body or the brain has been around since ancient Greece and these amazing portraits of Plato and Aristotle. But I'm going to be talking today about the scientific articulation of this concept, which came about from Richard Semon. And trigger warning for any of you out there that are very sensitive, I'm going to be talking about engrams. So Richard Semon was a scientist who defined the term engram, and he defined it as the enduring, though primary latent modification in the irritable substance produced by a stimulus. And he also defined this other term, basically it's a memory trace. He also defined this other term called ekphory, which is basically memory retrieval. And that is awakening the engram out of its latent state into one of manifested activity. Now, I love this definition, but I understand that some people find it kind of squishy. So this is my definition. So there's Semin looking forlorn. That's me looking fairly forlorn. I'm just really cold. And how I'm going to be using the term engram today is I'm going to be talking about the ensemble of neurons that are active during an experience that are somehow important in supporting the memory for that experience. And then ekphory, memory retrieval, is the process by which external or internal retrieval cues activate the engram and produce memory retrieval. So I'm going to show you some pictures of me throughout the ages, and you can see how tall I've grown in that time. So this is me in grad school. This is me. Next to me is my now husband, Paul Franklin, and our colleague, Stefan Kohler. We were together as graduate students back in the 1990s at the University of Toronto in the psychology department. And I was always really interested in the cellular molecular basis of memory. Paul was always really interested in the circuit basis of memory. And Stefan Kohler is a cognitive neuroscientist really interested in human memory. And we've been friends since graduate school and colleagues for life now. And every few years we get together and write some sort of review piece thinking about different ways of approaching memory. And one of my favorites is called Heroes of the Engram that we published in Journal of Neuroscience a few years ago. In it, we went through and we looked back, took a deep dive into the history of engrams and memory, and we identified people that we thought were really instrumental in our thinking. And we wanted to sort of talk about their lives and why they were really important to us. And we challenged everyone to go out there and say, okay, who's really important in your thinking? Let's take a deep dive and sort of appreciate them. So while we were doing this, we came up with a list of heroes, starting with Richard Semon, Carl Lashley, Donald Hebb. Lashley looked for an engram in the brain. Hebb, of course, LTP. Wilder Penfield probably found engrams in the human brain, arguably. Brenda Milner, of course. James McConnell, who studied memory and memory transfer in flatworms. And Dick Thompson, of course, who found engrams in the cerebellum. And while we really wanted to celebrate the life and the contributions of these folks. And while we were writing this, we were sort of joking with each other, it was 2017, that although these scientists were so important in our line of thinking, none of them would ever win science's highest prize, a Nobel Prize. Now, why is that? Well, this was 2017, and the majority of these scientists were dead. And one of the written rules, of course, of the Nobels is that you can't be awarded a Nobel posthumously. But Brenda Milner is still very much alive, 104 years old. But she violates an unwritten rule of the Nobels. She's a female. Let's dive into that a little deeper. So if we want to look at the Nobel gender imbalance in science and medicine, we can see that of the 600 Nobels awarded in sciences, only 23 have gone to women, and it's even bleaker for other groups that have been historically excluded from science. Of course, this is an old slide. If we update it, last year we had 7 new Nobels awarded. 1 out of the 7 was female, so we're still sitting around 4%. So I think we can do a better job of that. How can we do that? Well, I think this conference is one way of showing it. Invite women, cite women. So if you're gonna write a review saying that engrams are outdated and they're really crap, don't just cite Tanagawa, cite me too. So I'm happy to say, though, that Brenda Milner, along with Endel Tulving, won a Gairdner Award, which is like a Canadian Nobel, for their contributions to science. So I'm going to stop there on Nobels and just talk a little bit about Carl Lashley, which James McGaugh already introduced in the very opening lecture. So Lashley searched for an engram, that physical bit of the brain that seems to hold a memory, for about 30 years of his career. He read many, many important contributions to our understanding of the brain, but he could never find that bit of the brain that seemed to store memory. He lesioned out different bits of the cortex, and he could never find anything. So he published a very influential paper back in 1950, where he said that the engram was sort of everywhere, but nowhere in particular. The engram was very elusive. And from that point on, studies examining this sort of neuronal ensemble basis of a memory were largely abandoned around 1950. But then I started thinking about this, and some years later, 2010, I wrote a paper called, trigger figure. There we go. I wrote a paper called Continuing the Search for the Engram, where I thought I would try and update our findings on an engram by looking at a different type of memory. We're going to look at auditory threat memory and look at a different part of the brain. We're going to be focusing mostly on the lateral amygdala. So this work all started way back when I was a postdoc at Yale in Mike Davis's lab. And we were really interested in looking at memory and playing with viral vectors, which were just sort of being introduced at the time. So we collaborated with Eric Nestler's lab, who was across the hall. And what we did is we used viral vectors to overexpress this transcription factor, CREB, which had been implicated in memory in many, many invertebrate studies. We overexpressed it in the lateral nucleus of the amygdala, and we found that this enhanced conditioned threat memory. And this was a really, we thought a really cool finding at the time. There are many ways of decreasing memory, but not very many ways of increasing memory. So we were really excited about this finding until we looked at the brains of these rats. And what we found is, here's the outline of the amygdala in blue, that we infected only about 10 to 15% of these excitatory principal neurons in the lateral amygdala. And that sort of puzzled us. How could we get this large behavioral effect despite only infecting a pretty small percentage of cells in the lateral amygdala in just one brain region? So then we said, okay, well, what do we think an overall structure of the LA component of an engram would look like? Would it be that a very dense code with just about every neuron involved? Or at the sort of other end of the spectrum, might it be just one neuron or sort of like a grandmother cell involved? And of course, a lot of findings suggest that the code might be very, very sparse. So we said, okay, if it's a sparse code, maybe these neurons that we've infected that have increased levels of CREB are somehow preferentially biased to become part of this pretty sparse code that makes up an engram. So we set out to sort of test this hypothesis. So we figured that, okay, if the neurons that are overexpressing CREB are somehow really critical to this engram supporting this auditory threat memory, that they would be active following memory retrieval. And that's exactly what we saw. So here's neurons, the ones that we've randomly infected with our viral vector, overexpress CREB. And when we look after we test the mice, these neurons are more likely to be active. So we're just going to call this a proxy of an engram neuron, or a neuron that's really important in this engram. So this is what happens when we overexpress CREB, but CREB does so many different things. And then we tested many different ways that CREB could be affecting this bias. How is it that these CREB cells are outcompeting their neighbors? And we found that the common denominator was that CREB increased excitability, and that we tried a bunch of other ways of increasing excitability of these cells, and it all seemed to bias them for recruitment into the engram supporting this auditory threat memory. So then we did the opposite experiment. What if instead of increasing the excitability of a small population of cells, what happens if we decrease the excitability of these populations of cells? So we did this using two different ways of decreasing excitability, and what we found that memory was normal, because the code is very sparse, and these cells were actively excluded from the engram. So this allowed us to come up with the following sort of model. We figured that eligible lateral amygdala neurons were going to compete against one another for allocation to this sparse engram supporting auditory threat memory, that the one neuron in the middle outcompetes its neighbors because it's more excitable. So then we said, okay, if this is the case, if this neuron in the middle is outcompeting its friends because it's more excitable, and this neuron is really key to the engram, one way to test that would be to kill just this neuron, see what happens to the memory. And as a control group, we'll kill another bunch of neurons that we don't think are important in this memory and see what happens. So we think this is actually the experiment that Lashley was trying to do many years ago, but just didn't have the tools available at the time. So here we took advantage of a really cool way of killing neurons. We're going to have two groups of mice. We're going to overexpress CREB, train mice on this auditory threat conditioning, and then kill these cells. And then, in another group, we're going to kill a random but the same number of cells, but cells we don't think are really important in the engram, and see what happens to memory. So first, we're going to test mice There, the cells are killed. First, we're going to test mice before there's been any killing, and memory is normal in both groups. Then we're going to kill the cells just overexpressing either CRAB or a random control protein, which is GFP, and we only see a memory deficit when we kill those cells that were overexpressing CRAB before the time of training, rather than a similar number of just control cells in the other group. So this is a picture of Asim Rashid, an incredibly hardworking research associate in the lab. And he said, well, this is a really cool experiment, but killing is so 1990s, we have to use all optical tools. So what he's going to use is a very cool vector that allows us to manipulate the activity of the same population of cells with different wavelengths of light. So this construct is NPACY, but when I look at it, I might be a little bit dyslexic, I see P Nancy, and the folks in my group are just so lovely, they never correct me, and now everyone calls it P Nancy. So we have like this collective thing going on. So P Nancy expresses both channel rhodopsin, which is going to increase activity of these cells when we shine the blue light. But when we shine the red light in the same population of cells, we're going to decrease the activity because it's going to hit halorhodopsin. So we're going to, blue light increases the cell's activity, red light silences these cells, and the two wavelengths of light don't interact. So this is what Asim did in the experiment. So again, he's going to express this construct, channel rhodopsin and halorhodopsin, in the same random population of neurons. He's going to shine the blue light right before training to try and increase the excitability, try and recruit these cells into the engram supporting this memory. Now, how does he know that these cells are really important in the engram? Well, he's going to test the mice under two conditions. Once, under no conditions. The second time, he's going to silence just these cells he's tried to allocate into the memory trace, and he's going to see what happens to memory. And when he does that, in the red light negative condition, mice show high levels of memory, high freezing. When he turns these cells off in another test, everything's counterbalanced, we see a decrease in memory. So decreasing activity of just these cells seems to impair memory. In a control condition, he's not going to do anything to cells. He's not going to try and allocate them to the memory trace, but he is going to turn them off during a memory test, and he sees no effect at all. So together, we think these experiments, these results are suggestive of that neurons with increased excitability before the time of training become necessary components of an engram supporting that threat memory. So this is Sungmo Park and Emily Kramer. Emily's a grad student in the lab, Sungmo is a postdoc in the lab. And they said, okay, this is evidence that these cells are necessary for expression of this memory. What about sufficient? So of course, the entire brain is important, but what we're going to try in this experiment is to do a fun trick. So this is actually based on some beautiful work that Steve Amiris did when he was way back when he was just a lowly grad student. What we're going to do is we're going to allocate these neurons into the memory trace by shining blue light. And then during our memory test, usually what we do is play the tone. We think what the tone does is reactivate the engram. The mice retrieve this memory. So we're just going to cut out the middle person here. So instead of playing the tone to reactivate the memory, we're just going to do it optogenetically. So we're going to test mice in a novel neutral chamber. They don't do anything. We're going to turn these neurons on. We're going to try and get the mice to show us that they're expressing something similar to that memory. So we think this kind of gets back to what Semen was talking about when he described ekphory, awakening the engram out of its latent state into one of manifested activity. So maybe he was a little bit Nostradamus. We can attribute optogenetics actually to Semen. I don't know. So this is what happens. What we do, we put the mice in the chamber. They don't freeze very much. We turn on the blue light trying to reactivate these neurons. The mice show us freezing. We turn them off. They go down, turn them back on. We see freezing behavior. And we only see this when we allocate these neurons, when we train them. If we just do this in random populations of neurons without training, we see no increase in freezing behavior. So I started off showing you a picture of me, Paul Franklin and Stefan Kohler, my colleagues way back. This is us updated, and the latest, I know, we're all looking, I think I've grown a bit in that picture. And we wrote another review a few years ago called The Neurobiological Foundation of Memory Retrieval. And we're really going to focus now on ekphory. So memory retrieval has been always a very big, important topic in the cognitive literature, but maybe not so much in the neuroscience literature. So we're going to focus on a really nice finding from Endel Tulving and colleagues, where he found that there's the memory trace has two different states. It can be available, so the memory can be there, but not currently accessible. It's like the tip of the tongue phenomena. You know something. What is that person's name? Yeah, Tom Cruise, whatever. So the memory is there. You just can't access it at the time. And Tulving and colleagues came up with a beautiful set of theories based on this finding, and they called it the encoding specificity hypothesis. And that suggests that the congruence between the encoding and retrieval conditions really contributes to memory success. And my favorite illustration of this is this wonderful finding on folks that are really good scuba divers, where they taught them a word list and two conditions. One, they trained them underwater and got them to recall underwater, and they could recall it very well. Or they could train them on land, get them to recall on land very well. But when they mismatched the conditions, when they had them learn the list of words underwater and have them recall it, they don't remember it so well. So the memory is still there. They just can't access it because the encoding and retrieval conditions are a little bit mismatched. So we wanted to check the sort of engram encoding specificity hypothesis because we think engrams are very important in memories. And we sort of updated this to say that, okay, high congruence between encoding and retrieval contributes to successful memory recall because it efficiently reactivates the supporting engram. So the engram is still there and available, but sometimes it's just not accessible by certain retrieval cues. So in our sort of very basic auditory threat conditioning experiments, we have a mouse. This is how the memory is encoded. If we give it very similar conditions and ask them to retrieve the memory, it's a very good match. We get high levels of engram reactivation. We get very good memory. If we put them in a different context and we give them a different tone to retrieve this memory, it's not very good memory mismatch conditions. So it's very mismatched conditions. We don't get very good memory recall. Even though the memory's still there, we're not reactivating the engram, we're not causing memory retrieval. So we decided to do a bunch of different experiments trying to change up encoding versus retrieval conditions. And the one that we started with, of course, is, now I don't know if this was my mom or an old professor that used to tell me that if you study for a test drunk, you should take the test drunk. Actually, that sounds more like my mom. Anyway, so we didn't use single malt for this, but we did do that experiment, which I've been dying to do since I was a child. Everyone has their childhood dream, which was to train mice on auditory threat conditioning, either under the influence of ethanol or not, and then test them either under the influence of ethanol or not. And we want to look at this behaviorally, and we also want to look at the degree of engram reactivation. So we're going to use a RAM tag system, which Elena talked about yesterday, to tag the neurons that are active during the encoding event. And then we're going to use CFOS to check out the neurons that are active during our test event. So we're going to look at the number of neurons, engram neurons, that are tagged at the training event that are also active at the test event. So first, let's look at behavior. In our match conditions, so saline, or ethanol, we see high levels of memory. But when the conditions are mismatched, so trained under saline, tested under ethanol, or the other way around, we see decreased memory. Interesting. But this really replicates a lot of literature out there about state dependency. But when we look at engram reactivation, we see a really similar profile. So high degree of engram reactivation under the two matching conditions, even though one is trained and tested under ethanol versus the mismatch conditions. And when we look at the individual mice, we see a pretty high correlation between the degree of engram reactivation and the amount of memory, or the percentage of time that mice spend freezing. So that was one way we manipulated encoding versus retrieval conditions. And another way is by just presenting the mice with different tone cues to recall the memory. So here we play 1 CS, so it's a 2.8 kilohertz tone, pair that with foot shock, and then we ask different groups of mice to try and recall this memory using the exact same tone, a similar tone, or a different kind of sound, so pips. And here we're going to visualize the engram using our allocation strategy. We're going to again look at neurons that are positive for FOS as neurons that are active following memory retrieval. And what we see, which is in line with what many people have seen for many years now, is that mice show better recall when the training condition matches the test condition. So we use the same CS tone. A similar tone, we get a little bit of memory retrieval. A very different sound, we get very little memory retrieval. But what's really cool is that the degree of engram reactivation followed this profile. And again, we saw a pretty good correlation in individual mice. So these are the 2 ways that I'm going to talk about that we tried to match or mismatch encoding and retrieval, but we actually did many other experiments. So we used different ways of visualizing an engram. So I told you about the RAM tag. We also used trap2 mice, and we also used our allocation method with optogenetics. We changed match versus mismatch conditions on the encoding versus retrieval side with ethanol, different types of tones. And we also used internal cues. So we did optogenetic activation at different frequencies, and mice can tell these apart, which is very cool. I have no idea how they experience that, but they can tell them apart. And what we found is when we looked at the overall engram size, it didn't vary with the amount of memory recall, neither did the number of cells active during test. But across these very, very different conditions, 3 different ways of visualizing an engram, we got this lovely correlation. So we sort of stopped doing our engram encoding specificity hypothesis, and we look forward to doing some other fun ways of manipulating coding versus retrieval conditions. So then we said, okay, this is all very cool, but we're talking about allocation, but what might the function of allocation be? So we and others found that neurons that are more excitable are more likely to be allocated into an engram supporting a memory. But even after they've been allocated, after the training event, these cells remain more active than their neighbors. We've seen that, and some really lovely work from Michele Pignatelli's seen that from Susumu Tanagawa's lab. So again, asked them in his incredibly hardworking ways, said, okay, if these neurons are more active, what would the functional implication for that be? So he said, okay, if before event 1, these neurons are more active, and they remain more active for about 6 hours, 3 hours, depending on which brain region we're in and what type of training conditions. If a second related event happens within this 3-hour period, those cells that were active during event 1 would also be active during this second event. So they would be also co-allocated to an engram supporting these 2 memories. We call that co-allocation, and we think that this links memories, so that the memories become very linked. You think of one thing, you automatically think of the other thing. But Krebs has this very cool way of turning itself off. It recruits inhibitory isoforms like ICER so that these cells become refractory about 24 hours later. So cells that were once active are now less active, such that if the second training event happened now 24 hours after the first training event, the cells that were allocated to the first memory are not allocated to the second memory. We call that disallocation, and we think that allows mice to remember things separately. So then Assam wanted to check this by playing around. Is it possible to optogenetically overcome this? So what he wanted to do is allocate neurons to an engram supporting the first memory, and then artificially disallocate these neurons to a second memory that happens 3 hours later. So normally these 2 memories are linked because they're co-allocated. Well, what happens if he turns these cells off right before the second memory? Can he separate the memories? And it turns out he can. Then he wants to say, okay, if the 2 training events are 24 hours apart, we get different populations of neurons, disallocation. Can he artificially co-allocate them by simply reactivating the cells optogenetically? And sure enough, he can. So he came up with the theory that allocation is a time-limited, competitive process that serves to integrate or separate memories based on temporal context or maybe other things. So if we have an array of neurons, if one event happens and it's encoded by neuron A, B, C, and D, and E, a short time later, a similar population of overlapping neurons is set to encode event 2. Through a process of pattern completion, you remember one event, you automatically remember another event, and if a different event happens a long time later, a totally new population of neurons are encoding the second memory. So we think that the function of allocation is to organize information in the brain such that memories may be functionally linked or integrated or separated based on timing or potentially contents. So we think that allocation is a process for generating schemas or knowledge based on overlapping engram neurons. So this is what we've been doing lately based on looking at auditory threat conditioning. And a few of us have gotten together over the years, and we want to write a review. And I have the title. It's called In Defense of Freezing. So you know that freezing is a defensive response, and if you've ever tried to submit a grant to some large agencies, they say, oh, we already understand everything about auditory threat conditioning, and freezing is a very low output behavior. It's a passive response. Animals just aren't moving. But before I had an opportunity to write this review, I was anticipated by this beautiful review that came out from Peter Dayan's lab, where he and colleagues talked about how important it is to think about freezing as a very active response. Having said that, we wanted to see whether excitability-dependent allocation occurs in other forms of behavior, because I can understand that people think that freezing is a pretty sort of low-cost behavior. It doesn't seem very directed towards something. So we wanted to see whether this allocation process happens in a behavior that is a little bit more sophisticated than generalized angst. So we did show this allocation process happens for a rewarding memory in the lateral amygdala. But then we wanted to say, okay, what if we sort of step outside our comfort zone and look at another brain region? So this is work done by Ying Wang and Jagrup and Josselyn Lau in the lab. And what they wanted to do is think outside the triangle. Yes. So what we're going to do is go back again into the old literature, and we're going to adapt a task made famous by Olga Milner. So this is a picture of Peter Milner, ex-wife of Brenda Milner, and a wonderful scientist in his own right. So of course, Olga Milner, 1954, discovered rewarding brain stimulation. So they put an electrode in the medial forebrain bundle of rats, and they would turn it on when a rat was in a certain part of a chamber, and even when the rat was no longer rewarded, the rat would go back to that chamber. So we're going to take advantage of that and do a sort of spatial task based on this. So what we have is a mouse in a square arena with these distal cues on the wall, and every time a mouse enters one of these reward zones over the different trials, it gets a rewarding zap. And then 24 or 48 hours later, we put the mouse back and we say, do you like to spend time in this target zone? The place where you used to get this reward. Here is a heat map of a mouse, and you can see the outline of the target zone. And sure enough, they do like to spend a lot of time in that area, so they have a very good memory of this. We calculate the time in this target zone versus other zones of the square that weren't rewarded. So they show us really nice spatial memory there. We don't see it if we put, instead of a square there, if the same distal cues, we have a circle there. They don't show this spatial memory. And if we take down all the distal cues, again, they don't show us a very good spatial memory. Okay, so now we have a memory task, and we're going outside the triangle, so we're going to go into the dorsal C1 of the hippocampus. And we're going to go back to our old friend, the P-Nancy vector. So we're going to say, is it possible to allocate neurons to encode an engram supporting this spatial memory? So what we're going to do is we're going to randomly infect a small population of excitatory neurons in the CA1 of dorsal hippocampus. We're going to shine the light before we do the training, and then when we test mice, we're going to test them either when we turn these neurons off or we don't. So we have separate groups of mice. All the mice, as you can see here, show us really high spatial memory in the circle, the target zone, the northeast corner, except the one with the arrow here, because we allocated these neurons and then we turn off just these neurons. They no longer are showing us this spatial preference for this place. In all the other cases, if we turn off a similar number of neurons, but neurons that weren't allocated, they still show us nice spatial memory. Okay, so Ying said, okay, Asim got a really lovely paper looking at this allocation and the two memories. I'm going to do the same thing. So this is what he wants to do. Two different memory tasks separated 3 hours apart. We think there's going to be co-allocation. We think that the two memories are going to be linked or generalized. Two memories trained 27 hours apart. We think there's going to be disallocation, and the two places will be remembered separately. So Ying did this with no intervention at all, just looking at behavior. He's going to train them to the northeast corner in the square, and then either 3 or 27 hours later, he's going to train them in a circle in the southeast corner. And then he's going to test them both in the square. Do you remember the target zone is up there? In the circle, do you remember the target zone is down there? And sure enough, mice that are trained 27 hours apart, you can see they show very high preference for the zones that they should be showing preferences for. But mice just trained 3 hours apart show a pretty fuzzy generalized memory. So they don't show a specific memory for the target. It's kind of a blurry memory for the two targets, but it's not for the west side. It's always for the east side. So then he said, okay, can I play the same optogenetic tricks as we did with auditory fear memory? So first thing he's going to do is look at the 27-hour memory. The mice remember them distinctly. They have a really nice memory for the square versus the circle, but he's going to try and disrupt this by allocating neurons first and then reactivating the same population of neurons 27 hours later. So this is what he's going to do, just activate the neurons first and then activate them again prior to training in the circle. And when he does that, you can see that the mice do not show a specific memory for the precise target zone. Instead, they show a mushy memory. That's a very technical term, but I feel I can use this because this is a learning and memory audience. So without the light on, at 27 hours, they have lovely distinct memories. They know to go to the northeast corner in the square and the southeast corner in the circle. But if he attempts to co-allocate or generalize these two memories by just shining the light, they show mushed up memory. So they do not show a specific memory, but the mice still like the east side, north and south. They just don't have a preference. So he's going to do the exact opposite experiment now. Typically, if mice are trained 3 hours apart, they have this generalized memory. He's going to try and disallocate this. So he's going to first allocate neurons to the first memory, but these neurons are also going to be expressing the inhibitory GI dread. So he's going to try and turn them off when he trains them for the second memory. And sure enough, that is what we see. If he trains them under saline, they have a mushy memory. They like the East Coast, but they don't separate out the two different memories. If he inhibits these cells using dreads, he can get now a nice spatial memory for both the square and the circle. So he got very excited. He now thinks he has a handle on some sort of spatial engram that seems to be underlying specific spatial memories. So then he wanted to try and play these two engrams off against each other. So what he's going to use is an optogenetic intersectional strategy. So he's going to use two different excitatory opsons. One that is excited by red light, called carmine, and one that's excited by blue light, called channel rhodopsin. So he's going to use an intersectional strategy with a not function. So if I am a neuron in the dorsal C1 of the hippocampus, I can be uninfected, or I can express carmine, red sensitive, or I can express channel rhodopsin, blue sensitive, but I can't express both. So we're going to have separate populations of neurons that express each of these two excitatory opsins. So what he wants to do is try and allocate the red light sensitive carmine neurons to one memory and the blue light sensitive channel rhodopsin neurons to the other memory. And so what he's going to do is just allocate the carmine neurons before the square training and allocate the blue neurons before the circle training. And then he's going to test mice in the match conditions. So if he plays the red light that he tried to allocate the neurons into the square, if he plays that during the square, sure enough, memory is normal, everything is good. But the mismatch condition, so when he tests them in the square, but he plays the internal circle light, mice show a mushed up memory. So we think that there might be some sort of mismatch between what the mouse is experiencing in the external world and the internal world. So that was really cool to us. And then he said, okay, can I do the ultimate experiment? Can I get the mice to change their behavior in a novel context to respond as if they are not in a novel context, but instead in the circle? So what he does is he puts them in a novel octagon, and it turns out that mice seem to think an octagon looks like a square because they're up in the northeast side, which is all fine. But then he plays the blue light, and the blue light were the neurons that he tried to allocate to the circle, so the southeast, and when he turns on the blue light, every mouse except for that one lone mouse goes to that quadrant. So that's pretty cool. It's as if they are remembering something. Exactly what they're remembering, I don't know, but this certainly looks like a real memory-guided decision type behavior to me rather than general freezing angst. So from that, I just want to conclude by saying that excitability-dependent neural allocation to an engram happens in several different brain regions. We're trying to tease out what brain regions it does happen in, what brain regions it doesn't happen in. It's not just freezing. We think it's important in many different types of motivated behavior. And we think that engrams can mediate very, very flexible behavior. It's not just you have an engram, therefore there is no flexibility based into the system. We think retrieval is a really important part of the equation that certainly we and maybe other people have also overlooked. So with that, I just want to give a shout out to my funders and my husband, Paul Franklin, and my junior collaborator, my daughter there, and a huge shout out to the lab. Here we are on our annual selfie day, and this is before the pandemic, this is during the pandemic. And oh yes, just to say that that symbol at the top is because I now identify as Métis. I've always been Métis, but now I'm making it a point to tell people about that. Métis is one of the 3 officially recognized indigenous populations in Canada, and I am proud. Representation matters, and I usually have 1 or 2 indigenous folks in the lab over the summer trying to show them that yes, anyone can be a scientist. So we had our annual selfie day last week, but I haven't put the picture together yet. But just with that, I'm going to say we are always looking for really fun graduate students and postdocs, and yes, you could be featured in our next selfie day. So thank you so much for your time and attention.